With that, I'd like to, welcome Dr. Brianna DeMiranda to SRF, and thank you guys all for being here, for listening to her talk, and then also to thank Dr. DeMiranda for speaking to us and telling us all about additional research in the field. As I mentioned before, Dr. DeMiranda is a professor at the University of Alabama at Birmingham in the Center for Neurodegeneration and Experimental Therapeutics. She received her bachelor's and PhD both from Colorado State University, and has a really fantastic, did her postdoc at University of Pittsburgh. She has a really fantastic history, publication history of, dozens and dozens of papers and some really great, funding experience, including from the NIEHS, as well as I was really excited to see some from the Michael J. Fox Foundation that we've been kind of talking about recently. So we're really excited to hear about some of that work as well. Dr. DeMiranda also has a really amazing, numerous awards, including the Kalat Endowed Scholar in Neuroscience, the Toshio Narash, I'm so sorry, I'm slaughtering these names, Narahashi Endowment Award, and a few different times. So she's really brilliant, accoladed scientist, and we're really excited to welcome you to SENS Research Foundation and to hear more about your work. So with that, welcome, and we will turn it over to you.
Thank you. That was a very, very kind introduction. I really appreciate it. And I'm really excited to be here with all of you today as well. And like I said, really thrilled to present some background information on our research that we do here, as well as presenting some data that is considered, in my opinion, it's sort of hot off the press, but it's kind of where we are hoping to go in terms of where we see some openings for how we can really assess these neurodegenerative mechanisms that we think are playing a key role in mediating neurotoxicity from environmental exposures. So just as a brief note, my background is in toxicology. I am what's considered sort of a classically trained toxicologist, and I have focused 100% of my research on Parkinson's disease, pretty much from the day I started my graduate work until today. So there's a lot to be said about environmental exposures in Parkinson's, and that's what I'll be presenting on today. But this is becoming an important topic and has been an important topic for a long time in other diseases. The video that's looping in the background of my title screen comes from a steel plant in Pittsburgh where I did my postdoc called the Mon Valley Works plant. This runs 365 days a year and is a not uncontroversial source of emissions of pollutants in the environment in Pittsburgh. And I include it in my talks because there's a little bit of an irony about studying environmental exposures. Training in Pittsburgh, and now I live in Birmingham, Alabama, which is considered the Pittsburgh of the South. It is also an industrial steel city. They're both called steel cities. And it's interesting to live in places where you're really studying the things that affect the environment that you live in. So as we like to say in our lab, we live our research and we know how important it is to really assess environmental exposures in all of health and disease, but especially brain health. So that's just kind of a little bit of background on kind of how I got to where I am now and how my lab is continuing to assess toxicant exposure in neurodegeneration. So I thought I'd just give a brief overview. I know most people know what Parkinson's disease is, so I won't belabor any specific point here other than to mention that Parkinson's disease is considered an umbrella term, and what has really been happening over the last I would say maybe 5 years is to try to categorize Parkinson's disease on a spectrum. So if you have Parkinson's disease, or what's sort of defined as the classic Parkinson's disease, you might have mostly motor symptoms. Those are the things that you probably think about when you hear Parkinson's, like the slow movement, rigidity, resting tremor. But there are also types of Parkinson's syndromes like Parkinson's disease dementia, where the motor symptoms onset just 1 year prior to cognitive dysfunction, and that those symptoms then progress at a similar rate. And then Lewy body dementia, or sometimes interchanged with DLB, so dementia with Lewy bodies, is a syndrome of Parkinson's disease where the cognitive deficits onset 1 year prior to or in addition to the motor deficits. And so all of these are considered neurodegenerative movement disorders. They are all under the Parkinson's spectrum, but they're very different. We don't know what causes somebody to get Parkinson's versus dementia with Lewy bodies. If hopefully by the end of what I'm talking about today, you'll think that at least the environment could play some role in that. That's certainly what we think. But, you know, just as a general sense of kind of how clinically Parkinson's is defined and how we're trying to address that in the laboratory. So in addition to the motor pathology, there are a lot of non-motor symptoms like GI motility issues, depression and psychosis are highly problematic in Parkinson's, and then of course dementia. It's more common in men. Men also exhibit different symptoms than women. This is something that people are studying a lot right now as well. And something that we study in the lab is sex differences in response to neurotoxicity. So could people who develop Parkinson's disease from an environmental exposure be differently affected if they are male or female? And then, of course, we do have symptomatic treatments for Parkinson's. You probably know about L-DOPA, but just important to note that there are no disease-modifying therapeutics. So Parkinson's, or what I'll probably refer to as PD for the rest of the talk, is a systemic disease. It affects much more than just the brain. It's also a very long disease. It takes 10 or 20 years in the prodromal phase before somebody actually comes into the clinic and is diagnosed by a movement disorders clinician. So that's sort of what this graph represents, is at this line here, time 0, when you've been clinically diagnosed with Parkinson's, you probably have the disease 10 or 20 years before that. And the most common symptoms that you might exhibit are non-motor symptoms. So constipation and REM sleep behavior disorder all precede PD, and loss of sense of smell is really important in the prodromal phase. Most people report that they lost their sense of smell before they were diagnosed with Parkinson's. So then when you are diagnosed, it's really difficult to treat because you already have lost about 50% of the dopamine neurons in your brain. And this then progresses as the disease moves on. So for example, those motor symptoms tend to get worse over time. And this is where we again start to see a lot of dementia, a lot of cognitive impairment, as well as other motor and non-motor symptoms that can be very problematic and hard to treat, like pain, for example, is very difficult to treat in PD. And most people with PD will tell you it's not the motor symptoms that bother them, it's things like pain and cognitive impairment that's really affecting their quality of life. Underneath the clinical symptoms, we have a host of brain pathology. The key pathology in Parkinson's disease is the loss of dopaminergic neurons from the substantia nigra. These neurons project to the caudate putamen in humans. This is the striatum in rodents. That's what I'll be talking about today. So the substantia nigra and the striatum. We see a really strong neuroinflammatory response, predominantly driven by the resident immune cells of the brain microglia. And then a really key feature of most Parkinson's is protein accumulation. And like any neurodegenerative disease, their key protein that accumulates in Parkinson's is alpha-synuclein. And so it's the alpha-synuclein misfolding and accumulation that's thought to be toxic prior to the accumulation into Lewy bodies and Lewy neurites, which is what we see at autopsy in the brain of individuals with Parkinson's syndrome. So just a little background on that. we study Parkinson's disease as toxicologists. I'm not the only toxicologist that studies this, though there aren't that many of us out there, it seems, these days. But we study this because Parkinson's disease incidence really exists on a spectrum. So you can imagine one end of the spectrum, you have fairly rare genetic mutations that are associated with inherited Parkinson's disease. This can be things like recessive genes like PINK1 or Parkin. Those are very rare. They're autosomal recessive, and they result typically in very early onset, like a juvenile onset PD. And then you have autosomal dominant inherited mutations like LRRK2 or GBA. These are associated with late onset Parkinson's disease, and they do not display complete penetrance. So again, even with a mutation doesn't necessarily mean you're going to get Parkinson's disease. On the other end of the spectrum, we have very clear record of environmental exposures that have led to people developing Parkinson's disease. A very classic one that you've probably heard of is MPTP. This is a synthetic drug that people injected and they developed a very rapidly onset of Parkinson's, but that's just a very rare example. There are also examples of environmental exposures that have led to the disease. However, most people sit in the middle of this spectrum, so they have probably low penetrance susceptibility genes combined with a lifetime of environmental exposures, and this is what's inducing the majority of PD risk. Okay, so just to make that a little more complicated, it's not just genes and environment, it's everything. So you could probably take this for any disease. This is not unique to Parkinson's, but one of the things that we talk about in the Parkinson's field is the neural exposome. And this is trying to encompass everything from all of your genetic risk, so polygenic risk score, all of your environment, including things like your exercise, diet, medications you take, and then adding on things like exposure to industrial or industrial byproducts or chemicals in the environment that can contribute to Parkinson's risk. And that's really what's driving disease, but it's very difficult to study these. So in totality. So we study them in each individually and try to keep this big picture in mind as we draw concepts from our research. I focus mostly on pesticides and solvents. I do a little bit of work in heavy metals, but pesticides and solvents are the environmental risk factors, at least that are classified as industrial byproducts that are most highly associated with Parkinson's risk. So Parkinson's disease is also growing exponentially. It's outpacing the rate of aging. So by the year 2040, we're going to double how many people in the world have Parkinson's. And this is at least in part due, we think, to environmental risk factors. There are a lot of different environmental risk factors. So as I mentioned, Parkinson's is mostly an idiopathic disease. It's considered about 85% idiopathic and maybe 15 to 20% genetic. Of course, all disease has a gene or environmental component. We just can't link any direct inherited genetic mutations to most of Parkinson's disease. And there are a lot of environmental exposures we can link to Parkinson's. And so it's thought that the environment contributes a strong risk factor. So this is a graphical representation of those industrial byproducts that might be linked to Parkinson's risk. Each one of these that are listed up here have evidence either in human epidemiological studies or in experimental studies in the lab that show either Parkinson's pathology or elevated risk for PD. The pesticides are the largest category, probably because of just how many pesticides there are out there and how many of them can cause Parkinson's disease. But it's also very easy to track pesticide applications. So we tend to know where we put pesticides. Most farms have to register what pesticides they use. They know how much they use and when they use them. So for example, in California, the pesticide registry is much stronger than in everywhere else in the country. And so we have a lot of really good data in California about where pesticides are applied. We don't have that for anything else really on this list. So the halogenated solvents, which is what I study in my lab, heavy metals, these are all things that might be put into the environment by industry, but we can't really track it very well. Air pollution is tracked pretty easily, but we don't necessarily know which components of air pollution per se are being released at any given time, although we're getting a lot better about knowing where air pollution is. And then I have in the corner here pathogens and head trauma. These are not necessarily industrial byproducts. You could argue that they might be influenced by human behavior. However, it's really important to note that they are important in Parkinson's risk. So there are links to certain viral infections, as well as the gut microbiome is important in Parkinson's, and then head trauma as well. So just to try to give you a broad picture of how many environmental risk factors there are for PD. And then after I tell you this, I'm going to talk the rest of the time about TCE, or trichloroethylene, because we think this is a really important risk factor for Parkinson's, and it's something that we use in the lab in order to test these mechanisms. So TCE is a very small molecule. It's 2 carbon atoms and 3 chloride ions, and basically it's a solvent. So it's used in industry to degrease things. It was used since the late 1800s. It's been used for over 100 years, and it's really good at degreasing things. So it's used in vapor degreasing. It's also used in as a chemical feedstock for Freon, so to make refrigerants for all of our refrigerators. And it's been used really widely, but what you can probably see from this map here is that it was reduced in use throughout the United States over the last I would say 30 years or so. That's because it was actually classified as a carcinogen. And so most places have reduced its use. It was not banned until potentially this year, where the EPA has suggested that we probably should ban TCE. But unfortunately, because it's chlorinated, it doesn't break down in the environment. And a lot of this TCE, so where you see all these green dots, that is where it's been released, and it gets into the soil and the groundwater, and it doesn't degrade. So where it's been released, it's probably still in the environment. And so until we remediate this, it's going to remain a risk factor for a long time. It's associated with elevated risk for Parkinson's disease in epidemiological data. So one of my good colleagues and friends, Sam Goldman, has shown really excellent data on solvent exposure risk and Parkinson's disease, including TCE. He did a twin study back in 2012 showing elevated odds ratio for Parkinson's risk, and then a really strong study that he published in 2023, earlier this year, looking at the Camp Lejeune Marine Corps base that is in North Carolina, where a strong amount of TCE and similar solvents were released into the water from dry cleaners on site between the 1950s and the 1980s. And individuals who were living on base have now a 70% increased risk for Parkinson's disease. And this is a very large study. Over 250,000 veterans were included in this. So really strong data coming out of Camp Lejeune. I mentioned it's hard to track where we put these chemicals in the environment. This is an example of where we can actually say we have some data from how much TCE was released and how many people were exposed to it. So I mentioned that TCE gets into the soil. The main way that you're then exposed to TCE is when it gets into the soil. It can either remain in the soil, or it can hit a groundwater aquifer where it travels in a non-aqueous plume, and then it off gases into residential and commercial buildings. And so the most common way you're exposed to TCE is through vapor intrusion. And so this is like a passive inhalation of TCE. And so you could just be living or working in a building, and you could just be inhaling this all day long. And so, you would assume you're inhaling really low levels of this. If you were inhaling a high level, you would probably experience some acute effects of TCE, like dizziness or headache. And that would prompt somebody to come in and measure your air and say, this TCE is too high, and we'll remediate it. But that tends to not happen with very low levels. So that's, we think, kind of a chronic low level exposure is the most common. You can also be exposed if you work with it. It's less common now to work with TCE, but for many years, people would work with it in its vapor phase, and you could just breathe it in that way. It's also a drinking water contaminant. So it can hit drinking water and get into residential drinking water. At one point, it was the most commonly sampled organic contaminant in purified drinking water. I don't know if that's true like in 2023, but it was up until the early 2000s. There are other contaminants now that I think outpace TCE, like some of the PFAS, perfluoroalkyl substances, but those are not organic. So I think wherever you're debating on what's contaminating your water at the highest levels, TCE is usually in there, but we're getting better at getting it out. So lots of opportunities for exposure, unfortunately.
I can ask a quick question.
Yeah.
Is there a way to measure TCE in the circulation or tissue biopsies to measure TCE in the human body?
Yeah, so you can measure TCE in blood or urine, and its metabolites as well. The major caveat there is that you have to have been exposed to TCE probably within the last month for it to be able to be detected. So you do metabolize it and excrete it, and it won't stick around forever. The metabolites won't stick around forever. I think there is some data suggesting that some of the metabolites might hang around for 6 to 8 weeks. There's other data suggesting that in lipid soluble tissues, so like fat biopsies, as well as breast milk, it might be able to be retained a little bit longer than that, like maybe on the magnitude of months. But if you were a person who wanted to know if you were exposed to TCE like in the past, that would be very difficult to do because we would not have any way of detecting it unless the exposure was somewhat proximal to when you're taking the measurement.
Right. So is there a study done to correlate the exposure and how long an exposure has to happen before you start developing symptoms of the disease? Like, are there any retrospective studies done on that?
In terms of like blood or plasma levels of TCE?
Or the time of exposure to TCE and then they start developing symptoms?
Yeah, that's a really good question. And the short answer is no. There have been no studies to do that. And the reason why is because we didn't know this was a risk factor for Parkinson's until very recently. Okay. So we don't have a lot of data on that. It's very hard to get cohorts of people who have a disease involved in aging and then also try to find out what they were exposed to previously. So the best way we track that is through retrospective data, like the people at Camp Lejeune. I talk a lot about this with people on ways we might be able to improve on that. But yeah, it's really hard to do. So it's a great question.
Thank you.
Good. Yeah.
Just that the Lejeune study, I just had a look at it. It's quite ecological. They're just comparing Camp Lejeune versus Camp Pendleton because there was widespread exposure at Lejeune and not at Pendleton, and so you don't get the individual exposure and individual risk. You get sort of a mass level, and so it's difficult to really draw a causal inference there. It's unfortunate.
It's true. We don't know, and people would have lived at Lejeune and Pendleton for different amounts of time, right? So it's hard to pull out, it's very hard to pull out those differences. And I think actually the fact that there was a significant risk for Parkinson's to develop from coming out of Lejeune is actually, it's quite impressive because of all of those challenges. And so what we're probably doing is underestimating that risk. That's my guess. There's just not enough power in a lot of these smaller cohorts to really get to the bottom of that. But yeah, these are problems that we think about a lot. I'm not an epidemiologist, so this is why I model in my lab TCE exposure, because it's very difficult to get at this in the human population. One thing to note, people in the military are already at higher risk for Parkinson's disease. So again, the fact that it was pulled out that Lejeune versus the control cohort of Pendleton was a higher PD risk is actually, again, probably even more so over the civilian population, which would have been at potentially a lower Parkinson's risk. So there's a lot to consider there. And probably more data to come on that, I'm guessing, especially as people age into the risk for Parkinson's disease from the Lejeune cohort. They're still quite young. They're in their mid 50s as an average age.
Yeah, and one complication with all environmental exposure is that there are several factors, several environmental toxins that may be contributing to pathology as well. So you may have exposure to high levels of mercury. That also is a factor. Exposure to paraquat, pesticides, like you said, those things. So they all may be contributing to pathology and may be different in different people. Like what I'm saying is cohorts may be exposed to different kinds of environmental toxins.
Exactly. Yes, that's exactly right. And so when you look at a single risk factor, like say TCE, you're probably missing a lot of if you had just a group of people who were only exposed to TCE, it'd probably be a much stronger analysis, right? So yeah, so it's hard to do. There are efforts going on right now to try to understand better about what an individual is exposed to at one given time. Again, there's still going to be snapshots in time. We can't go back in history and see what someone was exposed to. It makes it very challenging to do this kind of work. And I think one of the reasons why we don't have more data on how exposures influence our health, because it is really hard. And our scientific system isn't set up to measure exposure, right? We measure biologic variables. And so I appreciate how clearly and quickly you're seeing the struggles here, because this is something that I think is really, really important in addressing. So great conversation. And we can talk more about this as I go on too, because it's something that I work on a lot. So yeah, thanks for the good comments. And that probably is a good segue actually to how we model this in the lab, because we've worked a bit at refining exposure to be more like what happens in the environment. So I'm going to talk you through a couple different ways we've done this. So we've talked about how TCE is inhaled. It can also be ingested through drinking water. And so we've basically developed two model systems in the lab to do this. So the first is through oral gavage. We started doing this. Actually, this was right before the COVID pandemic. We started exposing rats to TCE via oral gavage, and we used a pretty high dose here, 200 mg per kg. But that was still the lowest dose anyone had ever used experimentally. But the difference between our first study that we did and the studies previously done was we used aged rats instead of young rats, because we thought that the aged rats would be more susceptible to neurodegeneration from TCE exposure, and they were. So we saw degeneration of the dopaminergic neurons in the substantia nigra, which is what you see in red here, marked with tyrosine hydroxylase, or TH, the enzyme that produces dopamine. So that's what's in red. And then there are terminals that project to the striatum. You can see on the left here, you can see the degeneration that's occurring there. So we saw a degeneration of these dopaminergic neurons, but we also wanted to test this via inhalation. So we developed an inhalation chamber and exposed adult rats. So these were not aged adult, they were a little bit younger, about 10 months old. So these would have been 15 months in the oral gavage exposure and 10 months old in the inhalation exposure. We exposed them to 8 weeks, 5 days a week, of 50 parts per million TCE in the inhalation chamber. And just for a frame of reference, 50 parts per million is half of the time-weighted average daily exposure limit that OSHA regulates for humans. So we gave them half of what you can be exposed to as a human over an 8-hour workday in the inhalation chamber. And what we found is that we saw a really potent loss of dopaminergic neurons. This is TH, it's just in chromogenic staining and not fluorescence. And then the loss of the terminals from these rats here. So we saw a lot more neuron loss in the inhalation cohort, and it's much more potent via inhalation. So I'll talk a little bit more about why that is in a minute, but we can model this pretty easily in the lab. Okay, so
how long after exposure you are seeing the neuronal loss?
How long?
Yeah.
Okay, so
exposure do you see that? Neuronal
how long? So it's 6 weeks after oral gavage and 8 weeks after inhalation.
Okay, so you continuously give them the TCE, whether gavage or inhalation, and after
Yeah, we've checked it 3 weeks with oral gavage, and they had striatal, like they just started to lose their striatal terminals, but they didn't have significant neuron loss until 6 weeks. So it takes almost that full time. And then with inhalation, we've done a few time points, but we have not. We did them in mice, and I don't have the data on when the neurons die in that just yet. So that's all. It's all brand new. We haven't been able to run a time course yet. It takes a long time to do these chronic studies, so it's very hard to get all that data. But yeah, so 8 weeks and 6 weeks is when we see neuron loss for right now.
And they develop some physiological markers as well by then, right? Like some behavioral markers of Parkinson's by then. Okay.
Yes, so we see loss of spontaneous locomotor activity at 6 weeks of oral gavage, and the image below is just an expanded image of the neurodegeneration that I showed you previously. So we see motor deficits in the oral gavage, and we see specific dopaminergic loss. So this is selective. So we don't see loss of other neuron cell populations in the basal ganglia that are affected in Parkinson's disease. We don't see loss of medium spiny neurons that are labeled here in green. This is total neurons in NUN in the cyan color, and then GABAergic neurons we don't see loss of in the magenta color here. So this is the TCE treated below. So it's selective for dopaminergic neurons, at least in the basal ganglia. But we do see a lot of other pathology. So in the surviving neurons, we see a lot of oxidative stress. So this is kind of giving us an idea of how these neurons might be dying. We see oxidative stress marked with immunohistochemistry markers like 3-nitrotyrosine, which is a peroxynitrite adduct onto tyrosine residues. You have a lot of tyrosine residues in dopaminergic neurons. And then 4-hydroxynonenal is a lipid peroxidation product. It's marked in white. And this is just showing that you have lipid peroxidation in the neurons that are surviving of TCE treated rats. So a lot of oxidative stress involved in the pathology caused by TCE in the brain. We also observe an expansion of reactivity of microglia, indicative of a neuroinflammatory response. You can see in the large panels at the top here, in vehicle animals, kind of a traditional amount of microglia. It's a little bit concentrated in the substantia nigra pars reticulata, which you see down here, just below the dopaminergic neurons. And then that's really strongly activated by TCE. We see when we look really close at the microglia that the individual micros are activated. You can see in the bottom panel in green that we see kind of an activated phenotype of the microglia, and that they are expressing an expanded CD68 protein in red. That's the marker of the lysosome of activated microglia. So they're actively phagocytosing debris, and that's likely because we're killing the dopamine neurons. You can see them kind of around the dopamine neurons eating what's likely debris of dead cells, or TCE may be activating them, and then they're in turn damaging dopamine neurons. So we see a lot of microglial activation. And we also see a really key pathology of Parkinson's, which is the loss of endolysosomal function. So the inability to clear protein effectively. You can see that in the top panel, just marking some cell surface receptors like RAB5, which is on the early endosome. Those become accumulated in dopaminergic neurons of TCE treated rats. Suggesting that these early endosomes are not maturing into late endosomes and then lysosomes. And then we also see a buildup of P62, which is labeled on cargo that's destined for the lysosome, so it's not getting properly degraded. We also observe a loss of LAMP1 in these dopaminergic neurons of TCE exposed animals, and that's marking the late endosome and the lysosome. And so just really overall, a loss of endolysosomal protein function, indicating that we're getting a disruption of probably macroautophagy. This is corresponding with the accumulation of endogenous alpha-synuclein, again, the protein that forms Lewy bodies. So you can see in red phosphorylated alpha-synuclein, which is associated with its aggregation and toxicity, total alpha-synuclein in green, and this is within dopaminergic neurons of the substantia nigra. So again, a key pathological hallmark of Parkinson's disease. So what we found from this initial study was that TCE exposure can recapitulate hallmark PD pathology. I just walked through each of the pathologies that I have listed up on the slide here. So then thinking about kind of how we wanted to address this moving forward in using some of our newer models, like the inhalation model, we wanted to address this idea of, it's certainly a problem that TCE causes, the dopaminergic dysfunction that occurs in the basal ganglia, but we were also really interested in whether TCE may induce cognitive impairment as well, and that might be influencing the type of Parkinson's disease that someone may may get, right? So is this more associated with something like Parkinson's dementia? There are some very, some sort of older studies showing that people who are exposed to solvents may have cognitive impairments later in life. Okay, so the way that we want to look at this was to look a little bit more at alpha-synuclein, because there's a strong correlation with alpha-synuclein and cognitive impairment. This is a very nice summary figure from a published paper showing that you may have accumulation of synuclein caused by numerous factors, and then the severity of that can lead to the type of cognitive impairment that an individual gets. So in order to model this, we used alpha-synuclein preformed fibrils, which are the accumulated protein that's made in a test tube, and then we inject that into the brain of adult rats. So we injected in this cohort a unilateral striatal injection of alpha-synuclein and preformed fibrils, and then we exposed the rats to our 50 parts per million TCE exposure for 8 weeks. And this is again for considered a chronic exposure plus the synuclein. And this graph at the bottom is just showing you how we calculate time-weighted average, which is just that it's 50 parts per million over an 8-hour workday. So these animals go to work in the chamber every day, and then we take them out at night and bring them home to their home cage. So the pathology that results from this is very similar to what we see with TCE alone or PFF alone. So you would expect a loss of striatal terminals in TCE animals. We see a loss of unilateral signal in the PFF injected animals. And then when we combine the pathology, it gets worse. That's not all that surprising. I think, you're taking two pathologies that cause neurodegeneration and we're combining them. But what we found was that it was fairly severe and significant. TCE definitely induces more neuron loss than synuclein fibrils alone. So this is kind of getting at that idea that a person may have maybe increased risk for, say, alpha-synuclein accumulation, but maybe not until an environmental exposure came along and induced that synuclein pathology or made it worse, that they developed Parkinson's phenotype. So in addition, TCE worsened the amount of alpha-synuclein that was accumulated in the brain of these animals. So in order to track preformed fibrils that are injected, we use the phosphoserine 129A syn. And you could see in the PFF injected animals in this third panel here, we just put a marker over this in the panel B, so you can see a little bit more clearly where the fibrils are. Expected spread of fibrils in the striatal region near where we injected and into the cortex, but it's much worse with animals that were exposed to TCE. So we're getting a lot worsened Lewy pathology in animals exposed to TCE. This is true in the dopaminergic neurons as well. We see much worse endogenous synuclein accumulation, as well as endogenous plus phosphorylated synuclein accumulation in dopaminergic neurons when we apply preformed fibrils plus TCE, suggesting that we're getting, again, expanded Lewy pathology with the combined exposure. And so then one of the questions we asked was whether this would make cognitive impairment worse. And so we did, for this group, we did novel object recognition test, which is typically suggested for use for spatial or working memory. And we found that while there was some reduction in animals that received the treatments alone, it wasn't until we had the combined treatment that we saw a significant reduction in that novel object recognition test, suggesting that, again, that we are seeing cognitive impairment in these animals that are exposed to TCE, but perhaps when their Lewy pathology sort of reaches a threshold. So what I've shown you so far is that we can potentially induce both motor and non-motor symptoms of Parkinson's disease from TCE exposure. This could lead to the development of Parkinson's disease, or perhaps maybe modify or influence what type of Parkinson's you get, influencing the phenotype, like cognitive impairment, based on your underlying risk factors. So I just want to briefly introduce the mouse model that's sort of the companion to the rat model done by my graduate student, Ashley Adamson. This is essentially the same thing, but we have to use higher amounts of TCE in mice because they metabolize it much faster. It's kind of convenient, however, because the human equivalent dose, if you do allometric scaling from rats to mice, are both 8 parts per million. So 100 parts per million of TCE in mice is about 8 parts per million equivalent in a human. 50 parts per million of TCE in a rat is about 8 parts per million in a human. So they come out about the same because they're much smaller, they metabolize it faster. We expose them for 12 weeks. In the inhalation chamber, same paradigm. We put them in the chamber for about 8 hours a day and then 5 days a week. Similar loss of dopaminergic neurons. We see loss of motor. This is actually catwalk data now. So this is published in our ToxSci paper. So if you would like to read more on this, there's more information on the system, the motor system and gait system that we used. But we see motor impairments, and we also see cognitive impairments in these animals, which is a little bit different than what we saw in the rats. So using Y-maze this time, we see that there's a reduction of exploration in the novel arm, again, suggesting maybe spatial or working memory has been reduced in these animals. And you can see that that's here in the blue. TCE treated animals, mice in this case, are exhibiting a reduction in cognitive function in the Y-maze. In the interest of time, I'm just gonna go to, let's see, the end of my talk. I only had just a brief summary just on, you know, how we think both inhalation and ingestion could be playing a role in TCE exposure, how it relates to, you know, different systems, you know, whether it's coming from the directly through inhalation and getting more directly into the brain, whether it's going into the gut and being metabolized by the liver, and also affecting the microbiome. So just kind of a summary to note that, you know, TCE is a systemic exposure and it affects many organ systems in the body. So we think that it's tied to Parkinson's disease through multiple mechanisms, one being things like alpha-synuclein accumulation, things like you know, direct toxicity to dopaminergic neurons, as well as potentially through mechanisms that might be considered acceleration of aging. So I just wanted to leave plenty of time for discussion. So just a brief thank you to the individuals of my lab and my funding sources. And I appreciate all of your comments throughout the throughout my talk today, and I'm excited to talk to you more.
Thank you so much, Dr. DeMiranda, for your work and that exciting talk. I will open it up for questions, both obviously here in Research Center, as well as our folks that are online.
Great.
Yeah, I have a question.
Yeah.
Can you hear me okay?
Can you hear him, Mari? He's
I can hear. Yes. Yeah.
Hi, I'm Miguel. I'm the recent research director here at San Information. So a question about the micrograph, the last picture that you showed when you were looking at the picture. Do you have any plans on doing any partial transcriptomics by any chance?
Sorry, would you mind repeating? I can't hear very well.
I was asking if you were planning on doing any spatial transcriptomics on.
Oh,
Yeah, on the tissue section, let's say there.
yeah. Thank you for that question. I would love to do spatial transcriptomics. I would love to do that. It's I think we need to characterize this just a little bit better to understand what's going on regionally, but in sort of the next phase of this, I would love to do that. And that's definitely something we're interested in, as well as single nucleotide RNA-seq, just looking at you know, various cell populations. But yeah, great. That's definitely where we're headed with this. Once we sort of define, I think, the model and show that we have, show that we clearly have senescence and that it's driving part of the pathology. So one of the things we're going to do is we're going to use senolytics to try to reduce that effect and see if we can change any of the pathology. But yeah, absolutely. That's something that I'm very interested in.
Yeah, I'm asking because based on what I was saying, right? So P16 is not the only senescence marker. And with the spatial transcriptomics, you can search for multiple senescence markers there, and you can potentially, yeah.
Yes.
And also understand if senescence is contributing to pathology, or is it a consequence of pathology? And I think
Yes.
this experiments will give you, but like looking at the kinetics of when do you start seeing senescence burning in the brain would help maybe answer.
Yeah.
some-
Yeah, no, that's a really good point. So that's actually one of the funded projects we have is to do that. And we're going to look at whether we can try to get at whether we can intervene and see if we can reduce that pathology by potentially using either by clearing senescent cells or by using a senolytic. So a very good point. And yes, is it contributing to pathology or is it a consequence? And I think we also have some time course data that we want to very closely analyze to see before we get loss of neurons, if we can actually measure some of these changes that are happening along these senescence pathways. So like you mentioned, other senescent proteins or proteins associated with senescent changes, like transcriptional changes. And so that's something that we really want to look at. And we plan to. I mentioned a lot of this is really early. I wish I could have all that data to show you. But I'm glad that those are also things that you've thought of that could be useful for this.
One interesting, I think, thing that you mentioned in one of your introductory slides was that male are more susceptible to developing Parkinson's than females are. I think this is something you could address in your model. Have you compared male and female exposure and see whether they develop similar pathology or?
Yes, we have. One of our sort of, I don't even want to call it a side project. So something we're really passionate about in the lab is sex differences in response to neurotoxic exposure. And we did characterize male and female pathology from TCE exposure. And we really interestingly do not see sex differences at the level of pathology. So dopamine neuron loss, inflammation, like the really key things that are causing like motor deficits, we don't see sex differences. What we don't have data on yet, and what I'm really interested to see is whether at a more sensitive level, like epigenetic regulation, transcriptomics, if we see differences there, like do we see differentially expressed genes in male and female animals? So something that we really want to do. We could probably do that all wrapped up into one study where we look at transcriptomics. But yeah, that's a really good point. Now, interestingly, other toxicants like rotenone and paraquat and manganese and these others, they do see really clear sex differences in animal models. So TCE seems to be a little bit unique in that one. But there's definitely sex differences in toxic exposures as it relates to Parkinson's disease. Yeah.
Do we have any other questions online? What's there not in the research center?
Oh, but yeah, did I mishear you when you said that when you were looking at different parts of the brain, you did see P16 positive cells in Dante gyrus? Is that?
Yes. Yeah. In the dentate, we see P16 positive cells. Yeah.
Huh. That is interesting. Thank you.
Yeah. More to come on that.
All right, well, if there are no more questions, I will release everyone back to the labs, and thank you, Dr. DeMiranda, again for your talk and for chatting with us about your work. Really appreciate it. So thank you again.
Thank you all. I really appreciate it.